What is an active pharmaceutical ingredient in raw biosimilars? Have you ever wondered what makes biosimilars for rheumatoid arthritis so effective? The answer lies in the active pharmaceutical ingredient, or API. In biosimilars used for rheumatoid arthritis, the API is a biologic substance that closely resembles the active ingredient found in an already approved biologic reference product. These APIs are typically large and complex protein molecules, such as monoclonal antibodies or receptor fusion proteins. They specifically target components of the immune system that contribute to inflammation in rheumatoid arthritis. Biosimilars are crafted to be highly similar to their reference biologics in terms of structure, biological activity, safety, and efficacy. However, unlike small molecule generic drugs, which can be exact copies, biosimilars cannot be identical because they are produced using living cells or microorganisms. This means they are highly similar, but not exact replicas. Importantly, there are no clinically meaningful differences in how they work or their safety profiles. In the context of rheumatoid arthritis, the APIs in biosimilars often include monoclonal antibodies or fusion proteins that inhibit tumor necrosis factor alpha, also known as TNF-alpha, or other inflammatory cytokines. Some examples of these biosimilars include those for infliximab, adalimumab, or etanercept. These well-established biologics target immune pathways to help reduce joint inflammation and damage. Let's break down some key points about the API in rheumatoid arthritis biosimilars. The API is a biologic protein produced by living cells, often using mammalian cell lines, through a process called recombinant DNA technology. These proteins are large molecules, usually around 150,000 Daltons for monoclonal antibodies, and their complex three-dimensional structures are crucial for their function. The manufacturing process for these biosimilars is highly controlled to ensure that the API matches the reference product in terms of purity, potency, and immunogenicity, which refers to the potential to trigger immune responses. While minor differences in inactive components or microheterogeneity are allowed, they must not impact clinical performance. Biosimilars share the same mechanism of action, route of administration, such as injection or infusion, dosage form, and strength as their reference biologics. Regulatory agencies requiring extensive analytical, preclinical, and clinical testing to confirm that the biosimilar API is as safe and effective as the original biologic. In summary, the active pharmaceutical ingredient in rheumatoid arthritis biosimilars is a biologic protein that is highly similar to the original biologic drug's protein. This design targets the immune system pathways that drive rheumatoid arthritis, ensuring comparable therapeutic effects and safety profiles. This provides patients with effective treatment options that can improve access and potentially reduce costs. Always remember to consult with healthcare professionals for personalized medical advice regarding treatment options.